Welcome to Nature Calls, conversations from the Hudson Valley. Our team's goal is to present science-based information about gardening and all things nature in New York's Hudson Valley. Hosts Jean and Tim, along with team members
Teresa, Linda, and Annie,
are master gardener volunteers for New York's Columbia and Greene counties. So if you're interested in gardening or nature, or nuggets of information about what's happening outside your door, settle in. Enjoy the conversation. Whatever the season, we have something to say.
Here's part 3 of our retrospective series on pests and pathogens with Master Gardener volunteers Dede and Jackie.
Hello and welcome back to this segment of Pests and Pathogens. We're Dede Terns-Thorpe and
Jackie Hayden.
We're Master Gardener volunteers with Cornell Cooperative Extension of Columbia and Greene counties. Today we'll be talking about rust. And no, not what happens when iron oxidizes, but a fascinating group of fungus diseases.
The mechanism that rusts use is unique. Most require 2 hosts from completely different families and alternate between them. The scientific description of them from the U.S. Forest Services is this. Rusts are obligate parasites that depend on living cells and their hosts. Initially, they produce structures that grow into the host's living cell, from which they derive nutrients. They require that the host species be less than a mile apart and have from 2 to 5 stages of their life cycles.
So why are they called rusts anyway? Well, it's a descriptive thing. Cedar apple rust makes bright orange growths on the stems of juniper and eastern red cedar in the springtime, and orange or red leaf spots on the apple or the hawthorn leaves. White pine blister rust makes orange-yellow blisters on pine stems and orange spores with brown hair-like projections on the currants or the gooseberries.
2 of the most common in our area are cedar apple rust and white pine blister rust. Let's start with the cedar apple rust. This has 2 stages, one in cedars or jupiters and the other in the ribes family, gooseberries and currants. In the spring, the galls produce bright orange fungal growths with projections. They look like tentacles, and they release their spores.
We've seen those. I always wondered what they were. The growths dry out over the summer and may dry out and rehydrate several times over a season. They only release the spores when it's wet weather, so infestations will vary with seasonal fluctuations. And where do the spores go? Well, the lucky ones land on trees like apple, hawthorn, and serviceberry. They don't thrive on any other kind of plant. You can tell they're there by the red to orange spots on the leaves. Red dots show up on the spots as the spores mature, and then little stubby tubes appear on the underside of the spots. These release their spores. The spores can only affect cedars or junipers, and they have to be within about a mile. Both types of spores are slow-growing, so your best protection is just to keep an eye out for the first signs. That is, the red spots on apple family leaves and little galls on the cedars and junipers. Galls are certainly easier to spot when they grow those bright orange aliens. It's rare that you'll need to use a fungicide. If you do, read the label and remember that these plants are all important hosts to pollinators, which will be killed by the fungicide.
The other big rust problem is the white pine blister rust. This is another invasive problem. Its origins are in Asia, but not directly. Here's an interesting story. From Asia, the rusts were established in Europe as far back as the 1700s in white pines from America. Then around 1900, European seedlings were brought back to America. By 1950, it was in almost all the commercial white pine regions. These are 5-needle pines. Pine species that have their needles in pairs are not affected. The problem is that the 5-needle species, eastern white pine, western white pine, and sugar pine, valuable for their timbers, are the ones that are affected.
The infection is sneaky, too. Branch cankers develop and take 1 or 2 years to produce spores. Sometimes the needles near the canker turn bright red, called a blister rust flag, but by then the disease is well established. Often the cankers bleed resin from the damage of the cankers, thus the blister part of the description. The host supports the first 2 of the 5 stages required for a full cycle, the pycnial and e-shield stages. These then infect the alternate host, the ribes family of gooseberry and currants. The uridenial and the teleal stages must develop on the ribes, and the 5th stage is the basidio, which develop from the teleospores. After the ribes are affected by the spores from the pines, the underside of the leaves develop masses of orange spores, which infect other ribes nearby, expanding the exposure. These are followed by brown hair-like growths on the underleaf that give off spores that infect the pines, starting the cycle all over again. The cycle can take 3 to 6 years, depending on weather conditions, so it's always a potential attacker.
What can be done to interrupt the cycle? With the cedar apple rust, it can be kept down with manual controls like pruning. What methods have been used to control the white pine blister rust? Doesn't the fact that there are 5 stages give them more opportunity of control?
Well, one concerted effort was made to eradicate alternate hosts, ribes, the gooseberry, and the currant plants. The federal government banned growth of gooseberries and currants in 1911. And in 1966, states were allowed to opt in or out of the ban. New York didn't lift the ban until 2003. What's going on now?
There has been a move among the timber industry to designate risk zones and to prune lower branches of the pines so that the spores can't reach them. And as far as the ribes family is concerned, New York State is reviving the commercial growing of red and black currants in disease-resistant varieties. New York State Ag and the markets is spearheading the revival industry. Do you know that currants have 4 times as much vitamin C as oranges and twice as much antioxidants as blueberries?
That's all we have to say today. Thanks for listening, and we'll be talking to you again soon. Please go to ccecolumbiagreen.org for additional information. Thank you, and goodbye.
You're listening to Nature Calls, conversations from the Hudson Valley.
Hello, and welcome back to another segment of Pests and Pathogens. We are Jackie
and Dede,
and today we're going to talk about the infamous EAB, or emerald ash borer. It is a terrible invasive species that we believe to have arrived in wood pallets from Asian shippers. It was not lethal to the ash tree in its homeland because they have genetic resistance. Sadly, this is not the case in North America.
The life cycle of the emerald ash borer starts with the beetle laying its eggs between the cracks in the bark of a tree. The beetle, the scientific name Agregles plenipanes, is a beautiful emerald color, as you might assume. It's among a category of beetles called jewel beetles because of their beautiful gem-like colors. When first noticed in New York State, DEC began monitoring the presence of the beetle. You may remember seeing the purple sticky traps hanging in trees along major highways.
But the major effort is to slow down the spread of the insects was the ban on moving firewood from place to place throughout the state. This is because the insect is smaller than a penny, 3/8 to 5/8 of an inch, and can only fly half a mile from where they hatch. So the rule is to prevent humans from giving the killer a ride on a whole new feeding opportunity. Once the beetles hatch, they leave a distinctive D-shaped hole in the wood of the host tree. Before the larvae hatch, the only indication of their presence is an increased woodpecker activity in search of a tasty meal. They have spent a year in the larvae, sometimes 2 years, feeding on the tree. They leave the telltale S-shaped tunnels under the bark.
Sadly, if you see these tunnels, the tree is already dead or dying. In fact, an average tree dies within 2 to 4 years of infestation. None of the 4 types of native ash trees have any resistance to the beetle, and the damage financially is enormous. 8% of the forest population in New York State was ash, and it was a valuable commercial resource. Total timber income in New York State averages $9 billion. So loss of one entire type of tree is a disaster. Removal of dead trees is costly, and many were grown as street trees, so the local governments have the added responsibility of maintenance and removal.
As of January 2021, the USDA determined that the eradication of the EAB is no longer feasible. The termination of this program, trying to eradicate them since 2003, the total known spread of that time included 1,198 counties, 35 states. What actions remain include continuous monitoring of the spread, treating the certain few plantations with insecticides, and continue research attempting to isolate the factors that make Asian ash tree resistant. We can still help by not moving firewood from place to place. In fact, there are still regulations in place forbidding just that. Why make a problem worse if we can avoid it?
If you have an issue with ash trees on your property, DEC strongly recommends consulting an arborist. Only inoculations of particular insecticides are effective, and even that may be too late. Do not attempt treating the trees yourself until you have consulted CCE or DEC experts. Treatment is costly and must be regulated annually, so the trees must be in good enough condition to recover and thrive. If you insist on treatment, remember to consult. Follow the label, and remember, the label is the law. Thank you for joining us. Our website at ccecolumbiagreen.org will connect you to links with more specifics. Until next time, this is Dede
and Jackie.
Thank you.
You're listening to Nature Calls, conversations from the Hudson Valley.
Hi, and welcome to another episode of Pests and Pathogen. I'm Dede Thorpe.
I'm Jackie Hayden,
Master Gardener Volunteers with Cornell Cooperative Extension of Columbia and Greene County. Today we're stepping into the veggie garden to take a look at the squash borer. It's the larval stage of the native clearwing moth. We're interested in the control of this because the larval borers live inside the stem of all the plants in the squash family. Since that includes summer and winter squash and cucumbers, as well as pumpkins, gourds, and melons, it's important to know how to recognize the different indicators and prevent that infestation. If you wait until you see the wilting leaves, it may be too late for the affected vines. That means the worms have already eaten the inside vascular tissue of the plant and choked off nutrients to the fruit.
So how do you recognize these little guys? Start with the insect stage. They're what we call the diurnal species and the season moth. That means they fly in the daytime and they mimic wasps in appearance and for protection from predators. They also called the clearwing moths to distinguish them from the usual powder-winged moths. So you can watch for a wasp copycat with a wingspan of about 1 inch to 1.5 inches that show around your vines in late June. The goal in life is to lay eggs in the plant, usually around the stem and the stalks, around the base of the plant. The eggs are dull red and about 1/25 of an inch in diameter. If you find them, scrape them off. Always try to avoid insecticides because you could be killing many good insects as bad. In a couple of weeks, the borer hatches and drill into stems where the mission is to eat as much as possible. They leave the stems and the pupae. So if you haven't reduced the moth population or the pupae count, the larvae may reach full size and make cocoons. The cocoons overwinter in the soil and become pupae in the spring and turn into insects, thus completing the cycle.
Well, our goal is to interrupt the cycle whenever we can. Fortunately, there are a number of gardening practices we can use without resorting to chemicals. Start with floating row covers tucked down on the edges to keep the moths from reaching the plants, removing the cover when bloom begins. Stay vigilant and watch closely for eggs and little drill holes in the stems. The larvae are not good housekeepers, so you might see some frass, some bug poop, or ooze from the stem. It's early enough that you can go into combat. Either cut a slit in the stem and remove the worm, or take a sharp wire and thread it through the stem to kill the worm. Then push the cut stem back together and heap soil over it so it can make new roots. Sanitation is very important here. Remember, the cocoons can overwinter in the soil, so if you had borers one year, you should rotate the crop to another location the next year.
Also, be sure to remove the dead plants so that they don't harbor pests. Litter can be home for the next generation. There's a pesticide approved in New York State, but it's almost as much of a nuisance as the IPM methods we're just described. If you must use chemical means, read the label and remember the label is the law.
Thanks for listening. Please go to ccecolumbiagreen.org for further information. Thank you and goodbye.
You're listening to Nature Calls, conversations from the Hudson Valley.
Hello, and welcome back to another segment of Pests and Pathogens. I'm Jackie.
And I'm Dede Thorpe.
Today we decided to talk about fungus gnats and fruit flies. What's the difference? I always thought they were just different names for the same insect, and it showed up in different places.
Well, let's start with fruit flies. If you have elderly bananas, you might have fruit flies darting around. There are actually several species of fruit flies. The most common is the Drosophila melanogaster. If that name sounds familiar, they are very popular with scientists who use them extensively in genetic research. Fruit flies are indeed flies. Their size is between 1 and 2 millimeters, and they're kind of slow flyers. They tend to hover around the fruit they're attracted to. And Jackie, where do they come from?
They're most often brought into your house on the skin of the fruit you bought at the market. The eggs are invisible to the naked eye and have to have a terrific sense of smell. So the parents are in the warehouse and the orchards naturally, and of course they will lay their eggs anywhere they think they will survive. We bring home fruit from the market and the eggs are hatched. The newborn flies seek out the ripest fruit and they'll look for a feast. But they are different from fungus gnats. Do they look different?
Yes, they have fat bodies like many flies, and they're really small. They're easy enough to control without resorting to chemicals. You can just wipe down the skin of the fruit when you bring it home before you put it into a bowl. Or if you want to make a trap, you can recycle a plastic container with a lid, poke a few holes in the lid with a toothpick, and bait the trap with a little piece of banana peel. The little guys can't figure out how to get back out, and you've trapped them. About 24 hours usually catches the whole population. That's when you take the trap outside and release them, away from the house. Oh, and kids love this, by the way. Okay, so then what are fungus gnats? Aren't they flies too?
Yes, they are, but the name can be confusing. Gnats are related to flies, but are generally considered biting insects. Fungus gnats are not a biting insect. They belong to a whole genus of fly. The Latin name is Sayridae, and they are several genera and species. The ones that are interested in are the ones that you see around your houseplants. At first glance, they look like fruit flies, but they're distant cousins. But if you look more closely, they look more like tiny mosquitoes, 1/4 of an inch long. Their cycle is different from fruit flies, too.
We are most familiar with the little guys that hang around our houseplants. They look like really, really tiny mosquitoes, fly like gnats, and they're named after and seem to be attracted to light. It's like they want us to notice them. The flying stage isn't even the stage that causes the damage. It just makes us crazy. It's the larva form that lives in the soil that can be harmful. Fortunately, they rarely cause significant harm to the roots, and they're pretty easy to get rid of. There are yellow sticky traps. These are the bright yellow squares of cardboard that you hang on a little stick in the flower pot. The flies are attracted and get stuck on the trap. A full reproductive cycle can be as quick as a month with overlapping generations. It helps to make sure the soil dries out between watering. A top layer of fine sand or gravel can help keep the top of the soil too dry for egg laying. Too damp soil is their ideal habitat. If there is a persistent infestation, even with these corrections, there are approved chemicals. Use them sparingly, follow the instructions, and don't spray into the air. Be especially aware of them when you bring the plants indoors from a summer outside and when you purchase plants from a retailer. These are the opportunities for the wild population to lay eggs before the plant even arrives inside.
That's it for this segment. As always, ccecolumbiagreen.org will bring you to our website for more help. Thanks for joining us.
that concludes another episode of Nature Calls, Conversations from the Hudson Valley. We would like to thank
Sandra Powers
and Devin Connolly from Cornell Cooperative Extension of Columbia and Greene Counties for production support. And a special thank you to our listeners for joining us on this episode of Nature Calls, Conversations from the Hudson Valley. You can find links to any of the topics mentioned in this episode at our website at ccecolumbiagreen.org. Comments and suggestions for future topics may be directed to us at columbiagreenmgb@cornell.edu or on the CCE Master Gardener Volunteers of Columbia and Greene Counties Facebook page. For more information about Cornell Cooperative Extension of Columbia and Greene Counties, visit our website at ccecolumbiagreen.org or visit us in Hudson or in Acre. Cornell Cooperative Extension provides equal programming and employment opportunities